Okay. I'm also now at this time pleased to welcome back another one of our speakers that we've had previously. Dr. Nick Haley has, I think he's been a popular speaker here to say the least. And I think over the last couple of years we've had more of a relationship developed that we have a continual contact.
I can't quite figure out how to open this.
Oh, you're having problems. Okay.
There's not a. Is there a mouse or I can't even figure out.
Do you guys want my mouse again this year?
You got your mouse?
Yeah.
Oh.
I can't even see a mouse.
Alexis got her cordless mouse. We're going to get a cordless mouse here. No, moving it that way is the worst.
Normal is good for or maybe Frank, somebody. Not a good idea. It seems to have some function in neurological transmissions. That's where all what's the value of normal PRP? Well, there was a neurologist look at that one.
Well, he's up here. He keeps coming back, so I'll turn the mic over to him and let him go on his presentation.
Thank you, Harvey. So, I mean, I keep coming back because you keep inviting me, basically. But I just want to thank you for inviting me, for doing that. I just want to say from my perspective, as kind of an applied researcher in the United States, coming up here is always very refreshing. I think that you as an industry and Canada as a country is very forward-thinking when it comes to this disease. I know that you guys in the deer and elk farming industries have your, you butt heads with the CFIA and the environment people, but I just want to tell you that it's much worse down in the States. So, I think that you should be thankful for what you have. I also want to reiterate what Dr. Stasiak said. I'm sorry if I got your name wrong. I think that everybody needs to be working together on this disease. The target should be CWD. What it reminds me of is when my sister and I were little kids sitting in the back seat of the car, and Mom, she's on my side of the seat. That's what it seems like. I think there's a lot of conflict between wildlife and industry, and I think that the enemy in this situation should be the disease and not one industry or one agency. So the more that we can work together, whether we have differing ideas, I think the more successful we will be. I just want to touch on Dr. Knapper's presentation too. We talked a couple years ago. I think you did a great job of turning lemons into a little bit of lemonade. And there surely have been a lot of lemons in this research field, some of which I'll talk about for sure, and some of which we'll hear about a little bit later. But I think the important thing is to make use of the information that we find and try to advance our understanding of this disease. And I want to touch on using the natural models, so the deer and elk. I think that's important. I think mouse models are very important from a basic science standpoint, but there's a lot that isn't necessarily translating to the natural host. So some of these studies we have to do in the natural host as well. And I just want to say too that if we can't solve this problem within the fence, there's no way that we'll be able to solve it outside the fence. So studies like vaccine research, and that's great that we've had these conversations to maybe do these studies within the fence. That's great, because if we can't manage it in the fence, we'll never be able to manage it outside the fence. So I took a little time, but I'll try to get started here. We've seen these already. This was about when I started working on the disease about 12 or 15 years ago. All the really take-home message here is this disease is not slowing down. A little bit is because we're looking more, but a lot of it is because the disease is moving very quickly. Just in the past few years, and we've had 3 new states in the U.S. pop up. You guys had Quebec pop up this year. So you know, it's apparent that this disease is not slowing down despite our best efforts. You know, we just the other day I had to adjust this on my way here basically because Sweden came up with a positive case, so not slowing down. And you know, I'll kind of go over this pretty quickly because Glenn did a good job of talking about this, and I'll try to boil it down a little bit. You know, we're dealing with a gene that everybody has some variation of. The gene encodes a protein that's about 250 amino acids long, and you know, here's just kind of a stylized version of that protein. There are a bunch of different amino acids in that protein that help confer the shape of that protein, and this is the normal kind of layout of that protein, and then you know, one of the kind of key tenets is that the infectious protein has, you know, for all intents and purposes, an identical amino acid makeup. It's just that the shape of that protein that's different and is what makes it infectious. If the amino acid sequences of the host protein and the infectious protein are similar enough, you know, you can kind of think of it like a lock and key model. If those sequences are similar enough, you'll get CWD or CJD, and you can think of that key as fitting into the lock and unlocking the lock and causing disease. And just to kind of touch on the infectious prion protein a little bit more, there are different parts of that protein, like Dr. Telling mentioned earlier, and there's an unstructured region and there's a more structured region. And that structured region you can kind of think of like a Lego that you would play with as a kid. And those Legos kind of stack up on each other. And a lot of that stacking takes place in the more structured region of the prion protein. So now we'll take a step back, and I don't know if this animation went through. So, you know, now let's take a normal host prion protein and the infectious protein, but we changed just a single amino acid in the host protein. Sometimes that single amino acid change is enough to completely prevent infection in whatever host we're talking about. So I'll talk a little bit about sheep scrapie and what we know about potential levels of resistance in white-tailed deer especially. But sometimes, you know, just a single amino acid difference is enough to completely prevent disease in that host. And going back to the lock and key model, you now have a lock that doesn't fit that key, or a key that does not fit that lock anymore. And so I mentioned that I would bring this up. You know, I get a lot of conflict about what does the term resistance mean, and you know, you should say they're not as susceptible. And I think to myself that means the same thing. If they're not as susceptible, they're resistant. Or you know, we can use the word tolerant. That's fine too. But when you have a water-resistant watch, what does that mean? Some of you, I mean, I remember when I was a little kid, I had a water-resistant watch and I took it swimming. And you know, a water-resistant watch doesn't mean it's waterproof. So you need to consider that when we're talking about resistance to CWD as we know it right now. So, you know, just a standard definition: the impeding, slowing, or stopping of effect exerted by one material thing on another. So, you know, we can think of it as slowing or stopping CWD. You know, the hope is that we're able to completely stop it. But what we know right now is that as far as disease resistance in white-tailed deer, the best information we have right now is that we can't stop it, but we can do a really good job potentially of slowing it, and you know, we can have discussions about strain and all that sort of stuff too, and that's important. But you know, let's just kind of talk about this a little bit. So as far as resistance, you know, the best of what we know right now is that resistance means potentially prolonged incubation times. You know, maybe even beyond the normal lifespan of a deer. Reduced susceptibility, so they may be less likely to get the disease, you know, when we're looking at large herds of animals, or potentially even, you know, not likely to get the disease at all. So all of this is stuff that we're still trying to figure out with regard to disease resistance in cervids and CWD. So just to kind of go back to this kind of Lego model, what do we know about prion resistance? You know, a lot of the markers that have been associated with disease resistance in different species kind of fall within this sort of Lego region. So humans, 127, 129, those are two specific amino acids that provide some increased level or perhaps complete resistance to things like CJD or Kuru. Goats, there are a few different markers that provide varying levels of resistance to scrapie. Sheep, you know, some of you are very familiar with sheep scrapie and ARR sheep and BRQ sheep, so those are designations for different amino acids in the sheep prion protein that can to some level prevent infection with scrapie. And then deer, there have been 4 or 5 different markers that have been identified to be found in animals that are less likely to have CWD. And I'll talk about a few more, and just very briefly, that we've discovered just looking in the past 12 months or so. So just by looking in the past 12 months, we've identified 3 new markers that may or may not have some level of resistance that they provide in deer. Elk, and I'm sorry if you elk people feel like I've abandoned you, but you know, in elk so far we have basically one position that can be variable that provides some level of resistance, and you know, deer in my mind just offer a much bigger palette and potential of different markers that we can work with to some extent to learn more about resistance. So just to kind of bring everybody up to speed with regard to what's been done with sheep scrapie, most of you know, the work on prion resistance has been done in sheep and in mouse models as well. But as far as applied, you know, agricultural you know, changes, most of that work has been done in sheep. And as I mentioned, there are basically 3 amino acids that have been focused on in sheep: 136, 154, and 171. And different sheep breeds can have different amino acid markers at those positions, and even within breeds there's variability. So what's been done essentially over the past 15 years has been trying to change the genetic profile with regard to this PRNP gene in flocks of sheep, both in North America and Europe, to try to get them to a more resistant PRNP profile, so that we can reduce the prevalence of scrapie. In the United States, we've had you know, just maybe 1 or 2 cases of scrapie in the past year or so, and you know, there's been some work to try to identify whether those are the classical form of scrapie or the atypical form. But you know, over the past 15 years in the U.S. at least, we've reduced the prevalence of scrapie by 99% or more just by focusing on this one gene and changing the profile in the flocks in the U.S. And similar success has been found in other countries using this approach. So VRQ sheep are considered very susceptible to scrapie. ARQ or AHQ are somewhere in the middle, and in the sheep world, ARR sheep are what the sheep farmers are looking to produce. And the thought is, and I kind of have this color coded here, but VRQ sheep, for example, if they're homozygous for those variants, you know, they may go a year or 2 before they develop scrapie, before they develop symptoms. The ARQ or AHQ sheep, you know, they may go a little bit longer, 3 or 4 years before they start showing clinical symptoms and before they succumb to disease. ARR sheep, you know, as far as we know, they won't ever develop sheep in their or scrapie in their lifetime. But if we had sheep that lived to be 15 years of age or more, there's a chance that they could live long enough to develop. But you know, the average sheep doesn't live past 10 or 12 years of age. So you know, we haven't really seen in ARR sheep, like I said, the level of scrapie, the prevalence of scrapie has been reduced pretty significantly over the past 15 years or so. So there's a table that's been produced to kind of give everybody a formula for figuring out exactly what level of resistance each of these different amino acid changes mean in the sheep world. And so the VRQ animals homozygous for that VRQ, DRQ, they essentially have no level of resistance, or they're kind of the reference point. But just moving to, for an example, an ARQ, from a VRQ to an ARQ homozygous animal, they're 3 times as resistant. If you go from an ARQ to an ARR, now you're up to 13 times as resistant. And I want to kind of bring your attention to the terminology here. They're using the terminology resistance index, right? So even these ARQ animals, they're somewhat resistant, right? Or at least they're the resistant index. So they're not completely scrapie proof, but there's some level of resistance found in those animals, and so they're a little bit more tolerant to scrapie than the VRQ animals. So what about deer? So I mentioned there are a small handful of markers that have been identified in deer. We have 95, 96, 116, 226. White-tailed deer all have some potential variation at each of these points in the prion protein. So 96G will have a Q at 95, a G at 96, an A at 116, and another Q at 226. And then we have what's known as the 96S allele. It's essentially identical to the 96G. The only difference is at position 96, there is an S instead of a G. And then we can go down the list. The 116G is essentially identical to the 96G. The only difference is instead of an A at 116, it has a G. 95H, essentially the same as the 96G, except it has an H at 95. And then the 226K has a K at position 226 instead of a Q. So just single amino acid differences that have been identified in white-tailed deer. And all of them have been, in the past, through work on the wildlife side of things, identified as having some level of resistance, although it's been very difficult to tell because prevalence rates in those studies have been generally low, so we don't have a good feeling for how resistant they might be. The other important thing is, so a lot of attention has been placed on position 96, so both the USDA and the CFIA have tests that specifically look at position 96 and whether the animal is a G or an S. So outside of the 96S allele, what does every other variant of this protein in deer look like? So I kind of, hopefully you can see that. So outside of the 96, every one of the other alleles looks like a 96G if you're only looking at position 96. Does that make sense? So you can kind of think of it as, these agencies have sort of put blinders on, and they're kind of ignoring these other potential alleles that may have different levels of resistance than the 96S, for example, might have. So a few years ago, I wanted to, because we don't have a lot of data from the field about how resistant these different alleles might be, I wanted to kind of look at that in the lab. So we have this RT-Quick technique that I've talked about before that others have mentioned earlier. It's an amplification technique. You take a CWD isolate from an infected animal, and you mix it with protein that you've made in bacteria, and you can make those bacteria make a very specific protein. So you can make them produce the 96G protein or the 96S or the 95H. So first what I did was take a CWD isolate and mix it with the 96G protein. Very, very quickly, in a short period of time, we started to see amplification. So, normally this is about a 24-hour process, and within 4 hours we were seeing amplification in the 96G protein. So, if we know that the 96S protein is considered resistant, do you think it would take shorter or longer for the 96S protein to start to misfold? So it should take a little bit longer, and that's exactly what happened in vitro. So instead of 4 hours, now it was taking 6 or 8 hours to misfold. 95H, so we think of 95H as resistant, as somewhat resistant too. So do you think that that would take shorter or longer than the 96G? So a little bit longer, yeah. And the same thing, so what I eventually found was that the 226K out of all those different proteins took the longest to misfold. And when we, it's hard to kind of standardize this, but I took a canine protein, so dogs are thought not to get CWD and they're thought to be highly resistant. The 226K, the time that it took the 226K protein to misfold was about the same time it took the dog protein to misfold. So in my mind, I had a feeling that the 226K was going to be, one of the most resistant alleles out there. You guys in the deer farming world, you know about 116G. So, and you already know from the field data that I'll talk about in a little bit that they're resistant. So what would you think would happen with the 116G? Would it be shorter or longer than the 96G, for example? So you'd think it should be longer if they're resistant, but what we found was that it was actually pretty comparable to the 96G. So I wasn't too optimistic. But this is one reason why, lab data is important, but some of these things we need to do in the field. And so this, the 96S, the 95H, the 226K, they are all pretty comparable to what we knew about coming from the field data, but the 116G didn't quite match up. So in the field, it seems like 116G has some level of resistance, even though in this particular study, it didn't look to be resistant at all. So these are just some graphs of that amplification process. What I want you to see most importantly is this is the appearance of one isolate that's amplifying in these different proteins. This isolate was from Wisconsin, but we can take another isolate from Iowa, and these things might look completely different in terms of how long it takes them to misfold, in terms of which one is misfolding first. So I think that this RT-Quick technology could be very valuable when it comes to strain identification, for example. And that's something that I think is worthwhile looking at. So that's the lab data, but I think most importantly is the field data, the natural host data. So one important thing that I've been working on over the past couple of years is looking at depopulations from both the United States and Canada. So we had about 1,200 deer that were depopulated in the U.S. over the past 10 years, and 900 from Canada that we got very recently. I wanted to look at the genetic background of those animals with regards to the PRNP gene, and then go back and correlate that to their CWD status, not just positive or negative, but what stage of disease they're in. So we know that there are ways to identify animals in early stages of disease and late stages of disease. So I wanted to try to correlate their genetic background with the stage of disease they were in. So this is very raw data. There's a lot more work that needs to be done with it. But what I want you to kind of see is that we have 96G animals. They were much more likely to be infected than any of the other genotypes. And not only were they much more likely to be infected, they tended to be in later stages of disease. So what we would consider stage 3 or 4 or 5 of infection. So they had higher levels of deposition in the brain than some of the other genotypes. 96G, 96S animals, so animals that are heterozygous for 96G and 96S, they were about as likely to be found to be infected, but generally they were found to be in much earlier stages of disease, so stage 1 or stage 2. 96G, 95H animals, the prevalence seems to be lower in that genotype. And not only that, but the majority of the ones that we found were lymph node positive only, basically. And the same applies for 96S, 96S. The prevalence was much lower in that genotype, and I can't say that I've seen a 96SS animal that has been brain positive. They've all been lymph node positive only. So, using this, we kind of identified which of these are the least likely to have CWD, and which of these are if they are positive, are going to be in very early stages of disease as opposed to advanced stage CWD, and that's important. And I'll show you in a little bit. So, I'm trying to assemble this table like they have like I showed earlier with scraping with regard to ARQ, ARR, VRQ, that sort of thing. But obviously, the more information we have, the easier it is to assemble that table. Yeah, do you have a question?
Yeah, Alex, are there separations into stages? Is it arbitrary, or what criteria do you use to separate stages?
Yeah. So how do we define the stage? And that's it is subjective, but a 5 means there's a ton of it there, and a 0, a 1 means that it's lymph node positive only. So it's subjective, but it's fairly repeatable at least. Yeah, good question though. So we're trying to assemble this table to try and identify, relative to the 96GG animals, how much more resistant are the other alleles. And it's important to note that the 116G, I haven't found that in any farm deer in the United States at this point. I'm aware that it is in wild deer in Wisconsin, but I haven't seen it in any farm deer. And the 226K allele I haven't found in any Canadian herd, either the depopulated herd or the herds that you guys have been sending in. So that's kind of an interesting note. The lower lines of this table, all the lines of this table we need more work on, but we need a lot more data on the animals with 96S, 95H, 226K, 116G, in order to get a more accurate correlation between susceptibility and disease stage, that sort of thing. So just kind of going back to that scraping model very quickly, the thought is that 96GG animals live maybe 2 years before they start to develop clinical signs and eventually succumb. 96SS animals may live a little bit longer, and then we start to get out to these less common alleles. We don't have a lot of good information to say how long they might live, but there's a fair chance that some of them will live to be 7 or 8 years of age before they eventually die of CWD. And the thought is maybe that's long enough. What's the average age of a wild deer in U.S. or Canada? Anybody have any idea? What's the average age of a harvested deer in the 3, right. So if we can get them to live to be 6 or 7 or 8 before they start showing clinical signs, and the majority of harvested animals are 3, what is that? That's pretty important, I think. The issue is, when do these animals show up to be rectal biopsy positive? So chances are, if we start breeding towards resistant, it's going to be harder to identify them as rectal biopsy positive. Part of that reason is because they're less likely to have CWD and they move through the disease process more slowly. The other concern is, when do they start shedding? So, there's a lot of ideas about when they start shedding. I personally think, for example, they're not shedding in saliva until they become tonsil biopsy positive. They may not start shedding in feces until about the time they become rectal biopsy positive. So if we can keep these animals from, showing signs of infection in the tonsils or the rectal biopsies, we might be kind of preventing, or at least delaying the shedding of the prions so that they're not infecting other animals. But that's still a big question mark. We need to figure out when exactly these different genotypes are shedding and to what extent they're shedding. So we've spent some time between myself and Dennis Simpson, who's also doing some genetic testing in the U.S., the majority of genetic testing outside of one of the bigger companies right now anyways. Just looking at the variability of these markers in U.S. farm deer herds. Haven't done so much work in Canada, basically because I've only gotten 3 herds, and it's hard to make much sense of that. But it's just kind of interesting to see the proportion of GG animals compared to the other markers and how that varies across the United States. In Texas, for example, there's a lot of the 96S marker. It's one of the more common markers in Texas. It's even more common than the 96G marker. In other states, In Pennsylvania, for example, there are a lot of K and H markers, and what I think that just suggests is that these markers were probably independent mutations that showed up in one part of North America and just haven't had a time to kind of diffuse yet. So what can we do about CWD as far as genetic testing? We can continue genotyping animals to see what's out there. You know, we have 4 markers that have been reported before. We found 3 new markers just in the past year or so. I mean, the hope is that the more we keep looking, the more we might find something that, the more likely it is that we'll find something that's more resistant than what we know about already. We can try breeding resistant animals in CWD positive areas to see how much we can reduce CWD prevalence. We can discuss whether that's an appropriate action to take. We can breed resistant animals in negative areas to see how well that can be at preventing CWD from even taking hold in those states or provinces. So as far as continuing to genotype animals to see what's out there, I had a contract service for a while, but got to be, you know, I need to focus on the research. But there are contract services, including Dennis Simpson, for the PRNP sequencing. You know, we can test any sample that you want to send in and try to identify what markers the animals have. And we can do that on farm deer, we can do that on wild deer. We can do it on elk, we can do it on reindeer, we can do it on caribou or moose. As far as depopulations, you know, some of you have gone through depopulations before, but I think, you know, going forward, we need to work with these agencies to try to make sure that the genetic testing is done. We need it done down to a pen level, because all we have right now is, you know, I have 100 animals from herd A. I don't know what pen they were in, or, you know, all the positives are probably in one pen or a couple of pens, and so it's really hard epidemiologically to get a better understanding what's going on. You know, we essentially need more data. So the more data we can get from these depopulations, the more we'll be able to help you know, deer farmers and wild deer agencies, wildlife agencies to help come up with a solution for this problem. So as far as CWD positive areas and breeding different markers, I have a field project going on in Wisconsin. There's a ranch that has CWD on multiple properties. He's got a high prevalence property. 60 to 70% of the animals are infected on that property. He's got a low prevalence property where we've got about 5% of the animals coming off positive. And then he's got some breeding areas that don't have CWD yet. So we're looking at things like fawning rates, fawn health. We want to make sure that these animals with these markers are healthy, because there's anecdotal information that mule deer with one particular marker had bad hair coats, and what does that mean? I don't know, but we have, you know, we have deer with these markers that are top sires in the United States, and probably some that we'll eventually find in Canada that are top sires. So, I mean, the point is, we don't get to decide who's healthy and who's not. I don't care if they're scrawny. Mother Nature is the one who cares if they're scrawny or not. So, you know, we have animals with markers. They seem to be healthy, but we want to keep monitoring that to make sure that that's consistent. On this particular ranch, we've been looking at all the animals coming off and all the genotypes of those animals, and they're fairly aligned with what we found overall in depopulations. It seems like possibly the S allele is a little bit more resistant on this particular farm, so maybe there are regional differences in prion strains that might be more infectious to different genotypes, and that's something that we'll need to kind of actively monitor as we're working on these projects. But what I think is really interesting is that, you know, we just started working with him a few years ago, and within 2 years he's already cut the number of 96 G alleles in his herd almost in half, by basically, you know, identifying sires that have these genetic markers that he can incorporate into his breeding program, and actively breeding towards those alleles. Breed-resistant animals testing CWD susceptibility, so that's part of the Wisconsin project, but we're also working on a pen study with the deer farmers in the U.S. We expect to have fawns on the ground this spring, you know, a handful representing each different group of markers, so GG, GS, SS, KK, KH, all these different mixtures of markers, and the plan is to put them on a site that's had CWD, and follow them, collect samples, monitor their health, and try to see if we can identify when they start shedding and when they become rectal biopsy positive, and how long they may survive on that positive environment. And we'd also want to look at prion strain to make sure that we're not changing that agent somehow, which, from what we heard from Dr. Telling, seems like it's a fair possibility. We want to make sure that we're not changing it to be more infectious to humans than it already is. That's a potential possibility, but we could be changing it to make it less infectious to humans as well. So that study kind of looks like this. We have 10 groups of 6 animals and representing different genotypes, and we want to sample them every 4 months, biopsy, urine, blood, saliva, feces if we can, and then do a necropsy at the end, just to kind of get an idea of where that infectious agent was moved throughout the body. What's the catch? So we want to know why resistant animals are so rare, because they, outside of the 96G, they only make up anywhere from 10 to 20% of the animals out there. So is there something wrong with them? That's what we've heard. We want to keep monitoring that. How did resistance affect trace outs or live animal testing? Is this the best we can do? I want to kind of go over this quickly. I know my time is running out. Are some genotypes more susceptible to certain CWD strains? So why are resistant animals rare? Any number of reasons, which may or may not matter to farm deer. Maybe they can't do advanced calculus, but do they need to do advanced calculus on a deer farm? I don't know. In the wild, maybe they might need that sort of level of development. So again, in the Wisconsin Field Project, we're looking at spawning rates and just trying to make sure that animals with these markers are all, as far as we can tell, healthy. How will resistance affect trace outs and live animal testing? It's really unknown, because the USDA and CFIA are only paying attention to 96. So, most of these alleles look like they're 96G. So it's hard to say unless the agency starts changing their testing practices. We don't really know what's going to happen. But it is important to note that, because these animals, if they're infected, tend to live longer, part of the potential negative effects is that they'll be much more difficult to identify as positive through rectal biopsy. That might seem like a downside, but it's also because they're less likely to be infected with CWD. So these are all things that we need to take into consideration when we consider breeding towards resistance in farm deer. Is this the best we can do? We can, as has been mentioned, we can delete this gene altogether. It's been done in cattle, and we can completely prevent deer and elk from getting CWD. That may not fly very well with the wildlife people. So, the hope is that we can find some marker that provides complete resistance, that is waterproof and not just water resistant. So CRISPR, many of you have probably heard this word in the news. So this is a technology that can be used to delete genes relatively efficiently and effectively. But again, this is something that may not fly very well with, considering these are a large herbivore in North America and elsewhere, that maybe we don't want these genetically modified animals running out. Are some genotypes more susceptible to different CWD strains? So we don't really know this, but we're monitoring these properties because maybe we have to tailor what we think about resistance to a particular property based on what we're seeing coming off of that property, and we can make adjustments based on what we're seeing. So summary and conclusions: without things like CRISPR and other genetic engineering approaches, we're kind of limited to what we're able to find, and it's important that we keep looking to see what else is out there. We know a fair amount about CWD resistance, but we don't know how these markers impact farm deer herds, for example, and their health. But I think that several of these markers have the potential to be incredibly resistant to CWD. So whether you want to say that's resistant or less susceptible or tolerant, some of these markers, have the potential to be very resistant to CWD. And again, we might need to tailor what we're seeing to specific locations depending on what particular strain may be present in that location. And also I want to note that, when we're talking about resistance, even some primates are not what we would consider immune to CWD, so take that into account. So acknowledgements, I want to thank the North American deer farmers, some of you guys here in the audience, the deer and elk farmers, Whitetails of Wisconsin, people at the USDA, including Tracy Nichols, Bruce Thompson, and Justin Greenlee, students that I've had working in my lab, Gordon Mitchell with the Canadian Food Inspection Agency, who really came through and provided us some of those depops from the Canadian herds. We would have no information on the 116G without the help of the CFIA. And Wisconsin Department of Agriculture, and hopefully we can work with them on getting a pen study set up in Wisconsin. So, any questions?
Andy has a question.
Yeah, thanks. As usual, excellent presentation. Thank you for, again, taking the time to come up. You talked just a little bit earlier, you mentioned the various stages of CWD, and stage 1 is just in the lymph node and so on. Do you have a defined definition that industry could use or reference for the various stages?
So as far as a defined, staging process, that's with the CFIA and the USDA. I mean, they do have pathologists who can subjectively say, this is a stage 1, stage 2, stage 3, stage 4, stage 5. Yeah.
No, I understand that, and the challenge we have is when we try to get that definition from them, it's almost proprietary and they're not releasing it. So,
Yeah,
it's
so
really difficult as industry then when we speak to them and they speak to us and we have our negotiations and so on, to try to really understand what the stage is and so on.
So I typically have a slide, but I deleted that because I already took way too much time. But it's basically, we've seen some of these slides of staining in the brain and in the lymph nodes. And it's basically a little bit of stain, a little bit more stain, a little bit more, oh my gosh, it's everywhere, kind of staining,
So there's a level of subjectivity to it still.
There definitely is subjectivity, but a little is a little and a ton is a ton.
Yeah.
Yeah.
It's the in-between that we want to know more.
Right, yeah. And there are other concerns that I didn't specifically discuss. So there was a paper that came out recently that talked about the 95H protein and potentially being less resistant to proteinase digestion. And so there's a chance that we may be over-treating some of these, and so that's why, for example, 95H, it doesn't show up in the brain because we're over-treating it and we're obliterating it there when it's actually there. So, there's still a lot to work on, but I mean, the fact that some of these animals are found not to be infected or live much longer, I think that is a sliver of hope for me at least.
Chris?
You may not have the answer for this, but going back to the model of the sheep and that being the longest running model that they have, are they actually seeing genetically the animals that are resistant? Are they running into breeding? Because the concern is that if we breed towards this, we're breeding something out of them. What does the sheep industry experience? Are they?
There's always a concern for that, Chris. And at the start, it was the same thing that we're seeing here with the deer industry. There's something wrong with them. We don't want to breed with them. They have bad hair coats. They have bad racks. Blah blah blah. But I think that sort of tapers after a while, and there's always that chance. I don't know if any of you guys are dairy farmers or dairy farmed in your past, but there's a marker. There's Osborndale, Ivanhoe, I think it was the bull who passed down this genetic trait to like 80% of the cattle in North America, dairy cows, that caused them to have some disease. So it's certainly a possibility that we need to look out for. But right now we're in the very early stages of this disease.
But I guess my question was, has the sheep industry seen that?
They haven't. They haven't. Well, so there was one study that suggested that ARR sheep may be less fertile, but obviously we don't see sheep farms going out of business, so it's not significant enough to. And the truth is, we can't really relate that to that particular gene or not. I mean, it could be something very close to that gene, but It's something that we need to watch for, yes.
We'll take one more question and then we'll have a break for lunch.
So you said that the 226K has not been identified in Canadian herds. Geographically, that makes sense in the wild if it hasn't spread, but we've got lots of American genetics in our herds here. Do you have any reason or any idea why that might be with all the intermixing that's been going on?
That's a good question, and just talking to individual deer farmers yesterday and in the past few months, it almost always turns out to be just missed it by a hair, you know? This buck died before we were gonna collect semen from it, or this buck, we couldn't collect semen, I won't say why, couldn't collect semen on it for one reason or another. And it's just a matter of luck. And looking at these individual herds, it's been really interesting to see the genetics. I can almost always pick out a herd from Texas because there's just a ton of 96S animals in there. And you know, some of the herds from Canada had a lot of 116Gs, some of them had none. So it's just, you know, it's just random. Do you ever watch Price is Right and Plinko? And you know, it's just, that's how things go. So.
Okay, we'll cut it off there, and like I said, we've got the open forum this afternoon to catch up on anything we missed.